Yeah, I'll just. So,
Hello everyone, I'm Dr. Nisha Bhatnagar. I invite Dr. Neeru Thakral, our National Coordinator, ma'am, to introduce our today's expert. Ma'am, please do.
yeah, thank you, Dr. Nisha. So, good evening, everyone. It's our pleasure to have all youngsters here. We welcome you all our youngsters, for this Young Empowerment Program. This program is the dream of our President IFS, Dr. Pankaj Talwar and Dr. Shweta Mittal. So the motto of this program is to empower everyone, specially designed for the scientific program, for the research and everything to nurture. We are having this program every second and fourth Friday from 7 to 8. Fourth Friday, we are having followed by the quiz session also. So let's start without wasting with a wonderful time. So it's an honor and pleasure to invite the chairperson and to Dr. Ramaraju. So we have today expert Dr. Ramaraju with us. Welcome, sir. Welcome, Dr. Ramaraju, for this program. So we all know he's the founder and medical director, Krishna IVF Clinic, Visakhapatnam. He's visiting professor, University of Gießen, Germany. Adjunct Professor, Sri Ramachandra University, Chennai. Honorary Professor, PhD Guide in Biotechnology, Andhra University. Program Director, Krishna IVF Clinic, Extramural Research Center for PhD, Andhra University. Board of Studies Membership, Board Department of Human Genetics, Andhra University. Board of Study in Biotechnology, Board of Study DM Sri Ramachandra University, Chennai. Board of Study, Department of Journalism and Mass Communication, Andhra University. Advisory role also in the Board of Chairman, Merck, Mumbai. Member of Asia Pacific Advisory Board, Merck. Clinical Research Principal Investigator, Samsung, South Korea. So they have multiple publication in peer-reviewed international journal, contributed to multiple chapters in textbook. Award owner, is the recipient of the Delhi Telugu Academy Samkhya Bharat Gorakh Satkar 2004. Honorable Governor Rameshwar Thakur awarded Merit Certificate, Indian Red Cross Society. Indian Red Cross Society awarded Gold Silver Medal of Motivation of Blood Donor. Best External Chennai Partner Award, Samsung Research Institute. And Lifetime Achievement Award by Tapista 2023. So welcome, Dr. Rama, sir. It's our honor and pleasure to have you here, especially for your expert topic. So next, I invite Dr. Manjushri. Okay. Next expert, Dr. Anuradha. She is the founder, director, chief scientist officer, chief genetic consultant, Gene Tech Genetic and Genomic Lab, Hyderabad. PhD in human genetics, 27 years of experience in medical genetics. Designed multiple novel economic and application-oriented diagnostic tests. Postdoctoral work and experience in USA lab, Walkman Institute, Integrated Lab, and Lab Corpo of America. Young Scientist Award at Environment Mutagen Society, USA. Multiple paper publication in reputed journal, reviewer, author of medical book, chapter, and other higher educational courses. 350 plus CME lecture delivered at state, national, and international conference. Conducted 200 plus genetic awareness program. Designed world's first and most comprehensive NGS-based genomic panel for infertility and RPL, presented at multiple international platforms. So welcome, Dr. Anuradha. So today presenter we have Dr. Manjushri Kotekar. She's a senior consultant in IVF and reproductive medicine, ART Fertility Clinic, Mumbai. So yes, Manjushri, now you can share your screens. Himanshu, you can stop screen.
Good evening, everyone. So I first of all would like to thank you, IFS, for giving me this opportunity to present here today. I chose this topic because this topic has been baffling me in day-to-day practice, And thank you, IFS, for selecting such esteemed experts also who are experts in this particular topic. And this today's discussion is definitely going to help all of us in our day-to-day practices. So I'll start. So as we, can you see my screen?
Yeah, so topic
Yes.
we have effect of polymorphism in
Yes, FSH receptor polymorphism. So I have taken 3 papers, ma'am. Out of them, the first paper, uh, I'll start sharing this. I'll start the PPT, and then as the PPT unfolds, we'll see. So we all know that FSH is a glycoprotein which is secreted by anterior pituitary under GnRH control, and it is under the influence of activin and inhibin. It has got alpha and the unique beta chain, which confers the specific biological action to the FSH. The FSH receptor is particularly, it's a G protein coupled receptor with a long extracellular domain, a transmembrane part, as well as 3 short intracellular loops, 3 extra loops, and intracellular tail. The gene for this receptor is located on chromosome 2p21p16, and it's a single copy gene with 54 kb in length. There are 3 kinds of defects possible: mutations, which can be activating mutations or inactivating mutations. Polymorphisms, around 1800 single nucleotide polymorphisms of the FSH receptor gene have been reported in the SNP database. And the 2 best characterized polymorphisms which have been studied extensively are they are nicknamed as RS6165 and RS6166. 6165 is between ALA307 and TR, and 6166 is between SER680 ASN. And apart from that, there are 4 different isoforms of the receptor which have been noted, but the clinical significance is questionable. Now coming to the paper. This is a paper from Human Reproduction published in 2021. The effect of polymorphisms in FSH receptor and FSH beta genes on ovarian response, a prospective multicenter multinational study in Europe and Asia. The study question here was, does the presence of single nucleotide polymorphism in the FSH receptor gene and FSH beta subunit encoding gene, FSHB, influence the ovarian response in predicted normal responders treated with a recombinant FSH? And the summary answer was that the presence of SNPs RS6165, RS6166, and RS13942005 have a statistically significant impact on ovarian response. Although this effect is of minimal clinical relevance in predicted normal responders treated with a fixed dose of 150 units of RFSH. So it was a prospective, non-interventional study. They had taken patients from Europe as well as Asia. Patients from 3 countries were included: Spain, Belgium, and Vietnam. Patients doing their cycles between 2016 to 2019 were included. The inclusion criteria was predicted normal responders. Age should be less than 38 years, first or second ovarian stimulation cycle with a predicted normal response as defined by the normal ovarian rhizome markers. Patients with AMH less than 1.1 or when the AFC was less than 9, and PCOS patients also were excluded. Patients with untreated endocrine anomalies and those who were undergoing IVM were also excluded. All the patients were stimulated with a fixed dose of RFSH 150 units daily from either day 2 or 3 or 5 days after stopping the OCPs. It was a fixed antagonist protocol. No dose adjustments were allowed throughout the stimulation. Trigger was HCG 5K or 10K or 250 micrograms of recombinant HCG. Only GnRH agonists were used when there was a possibility of OHSS. OP was done 34 to 36 hours after the trigger. Blood sampling for the receptor was done prior to OPU, and all the blood samples were stored at minus 80 degrees Celsius until the time of genotyping. And with PCR technique, they had the genotyping was done for 3 SNPs: RS6165, RS6166, and RS13942005, and 1 SNP for FSH beta chain. And the lab staff and the clinicians were blinded to the results of the genotyping. So they compared for each polymorphism and the patient genotypes according to following outcomes. They compared the percentage of patients categorized as hyporesponders. Their definition of hyporesponse was total number of oocytes retrieved less than 10. Then they compared the total number of oocytes retrieved, and they compared two ratios: follicular output rate FORT, which was the ratio between the follicles that reached the pre-ovulatory maturation state between 16 to 22 prior to trigger, and to the number of antral follicles which were seen at the start of stimulation, and follicle to oocyte index, which was the ratio between number of oocytes recovered at the end of stimulation and the number of AFCs. They had calculated the sample size, and so their interpretation was that around 365 patients would be required. So they had taken 167 patients from Europe and 198 from Asia. The mean age of the patients was 30.49, mean BMI was 21.26, and the basal AMH was mean basal AMH was 3.8 with a AFC of 17. This table, in this table, we can see the causes of infertility. They were male factor, idiopathic, tubal, or others. But ovarian causes were not there because they were predicted normal responders. So when they had seen tried to study the prevalence of the genotype defects as per the continent, it was seen that RS 6166 GG was more frequent in Caucasian population than in the Asian population. For RS 6165 GG and the other FSHR SNP 1394005, AA was more frequent in Asian than Caucasian. And the prevalence of FSH beta 10 SNP RS 108-35638, the prevalence of homozygous status was very rare, and it was not included in the analysis, further analysis. When they studied the prevalence of hyporesponse, there was no significant difference in the prevalence of hyporesponse according to genotype, even when the data was analyzed as per the continent. For RS 6166, there was significantly higher prevalence of hyporesponse in patients who were heterozygous AG as compared to AA. And for RS 6165 and the other RS 1394205, there was no significant difference. When they studied the number of oocytes recovered, it was seen that there was a statistically significant difference in FSH RS6166 and RS1394205 in terms of the number of oocytes recovered. In this table, we can see this minus 1 point. This is the actual difference in the number of oocytes recovered, mean difference 1.59, 1.07, 1.47, and all that. But for RS6165, there was no significant difference in the number of oocytes recovered between the genotypes. When FORT, the follicle output recovery rate, was compared, there was no significant difference in terms of all the genotypes in all three kind of SNPs. And for follicle oocyte index, it was found that for the variant RS6166, there was a lower FOI for AG, the heterozygous state. And even for RS6165, there was a lower FOI for the genotype AG, which was the heterozygous state. So the conclusion was that there was statistically significant difference in ovarian response, statistically significant lower number of oocytes recovered in heterozygous patients for RS6166 and RS1394205, and significantly higher rate of hyporesponse in heterozygous patients for RS6166, lower FOI for 6166 and 6165. But the clinical relevance was minimal, as we could see in the table which was given, that there was the actual difference between the number of oocytes recovered was only 1 or 2 oocytes. So they have concluded that FSH receptor polymorphism testing is not recommended in expected normal responders, and they have further said that more research is warranted in expected poor responders. The strengths of the study are the stimulation parameters were fixed, though there were no dose adjustments allowed. The findings were blinded for the lab staff as well as the clinicians, and ethnic variations were included. They had included patients from European as well as the Asian background. But the limitations are only expected normal responders were included. So we need to exercise caution when we are trying to extrapolate this data to expected poor responders, because in that case, the difference of a single egg retrieval can be significant. And they had not performed any analysis for the variant in FSH beta, that is RS108-35638, due to the very low prevalence of homozygotes in the studied population. I have also taken this paper. I'll just go briefly. This paper was about FSH receptor polymorphism status and its impact on the live birth rate. It was from, hello? Yeah, it is a study from JHRS. It was a retrospective analysis. The aim of the study was to examine the pregnancy outcomes and the responses to controlled ovarian stimulation according to the polymorphism types. Inclusion criteria. I'm sorry. Okay. The inclusion criteria was patients getting treatment between 2018 to 19 were included. First, IVF transfers, IVF with ICSI, age between 18 and 40 years, BMI less than 30 were only included, and presence of both ovaries with a normal karyotype. Women who had conceived with non-IVF assisted technique, and women who had previous IVF transfers who had undergone only IVF and not ICSI, DOR patients, male infertility patients were excluded. Because it was a retrospective analysis, here there was a varied combination of agonist as well as antagonist protocols, and the stimulation was also either with RFSH or with urinary FSH. Doses were ranging between 150 to 300, and all the patients had undergone a fresh transfer either on day 3 or day 5. They had included 143 patients, and they did the PCR amplification technique. So they had only studied the RS6166 kind of polymorphism, and what they found was 14% of the patients from their studied group were homozygous natural, that is AA. 44% were heterozygous mutant, that is ASN SCR, and 41% were homozygous mutant, that is SCR SCR. When we saw the demographic and the medical characteristics of women between the three genotypes, there was no significant difference. Even the AFC, the basal FSH, and the basal E2, all the parameters were not significantly different. But when the treatment characteristics were seen, it was found that there was a significant difference in the peak E2 achieved on the day of trigger. It was lower for SCR group than the heterozygous ASN SCR group. Even the number of oocytes retrieved was less for the SCR group than the ASN SCR and the ASN group. But when they studied the treatment outcomes, there was no significant difference in terms of pregnancy rate per started cycle, ongoing pregnancy per started cycle, and ongoing pregnancy per embryo transfer, and the live birth per embryo transfer for all three genotypes. The limitations of this study was that it was a retrospective study, so there was a varied combination of all kinds of stimulations and all kinds of patients. The number of patients was also limited. Stimulation protocols were not uniform, and there can be racial and ethnic variation between the polymorphism, and this point was not included in the analysis. And this was a paper where our esteemed sir, Professor Ramaraju sir, was also part of the panel. So here some experts had come together, and they had kind of arrived at a consensus. They had given some statements which we can all rely. It was a Delphi consensus about the genetic variants, effect of genetic variants of gonadotropins and their receptors on ovarian stimulation outcomes. So I'll directly come to the statements which they have released. For RS6166, the SCR carriers require higher amount of gonadotropin during ovarian stimulation than the ASN carriers. The SCR carriers showed higher basal levels of FSH, and they produce fewer oocytes in response to ovarian stimulation. And one point which after revoting was approved by the experts was that the available data in literature suggests that probably SCR carriers tend to produce fewer metaphase 2 oocytes after ovarian stimulation as compared to ASN and ASN SCR carriers. And there is mixed evidence supporting an association between this ASN SCR variant and OHSs. The statements they have approved for RS6165 were not they could not arrive at the consensus of all the experts which had come to form this consensus, but after revote they have released these two statements that few studies suggest that the THR carriers of this RS6165 require a shorter duration of gonadotropin stimulation than THR/ALA and ALA carriers, and ALA carriers of this RS6165, which is in strong linkage disequilibrium with SCR RS6166 variant, produce fewer oocytes in response to ovarian stimulation than the other carriers. For this RS1394205, the consensus statement which was approved after revote was that limited data in specific ethnic groups suggests that AA allele carriers of this variant require higher amount of gonadotropin and produce fewer oocytes in response to ovarian stimulation than GG carriers. For FSH beta chain SNP, the data on an association of FSH beta with B cell levels of FSH and production of oocytes in response to ovarian stimulation are contradictory. This is the consensus statement. And for LHB and LHCGR gene, about which our sir had done a lot of work, the consensus statement was that limited data suggests that polymorphisms of these genes can influence ovarian stimulation outcomes and may represent targets for pharmacogenic research in ART. So I would stop here. I would like the comments of our experts. The guidance for us. Should we consider testing patients for this FSH receptor polymorphism in
Dr.
our
Manjushri,
normal?
I think the question should not be should we consider. I think the question should be whom should we consider? Because it's not for all. Where we should be doing this testing and what is the clinical significance of this testing? Because maybe Dr. Raju, can you please elaborate a little bit? I know that there are various tests are available. So my straightforward question to you would be number one, when should we decide that the patient should go for a gene polymorphism test? Number one. Number two, if we decide that the patient has to go for gene polymorphism, what test we should be advising? Should it be only particular genes, or it should be the whole set, or what else can we do about it? And number three, how much do you see the difference if you do go for these tests in the outcome?
The first thing in the
You can
whole
stop here now.
scenario,
Yeah.
in the whole scenario is we never define what is the starting dosage.
I also feel individualization is the key. You were saying that the strength of the study was that there was no dose variation. I would say that was the limitation of the study because the dose should be individualized. No,
Yes,
sir?
and because what happens is, first of all, is we never define what is the starting dosage, and we don't keep it uniform on a principle basis. In our practice, use the following parameters to plan the starting dosage: age, AFC or AMH. Third thing is presence of ovarian surgery, be it endometriosis. Then the third thing is the thyroid antibodies. The fourth thing is BMI. The fifth thing is collagen disorders. All these make an impact on the starting dosage. The second thing is we keep on changing the product, and we never document the batch number. And when we keep on changing the product, because not all HMGs or recombinants are exactly the same molecules. Each of them have a slight difference. We keep on switching from one product to another, and then in the process, we are comparing different sets of data sets.
Sir, can I please ask a question here? When you say that there is a difference in the product, is it true for even the recombinant ones which are produced in the lab?
Yes, ma'am.
Or, oh, that I didn't know. I thought there is a consistency.
There is less batch to batch consistency, but we cannot say it absolutely no in recombinant.
Because what happens is the ferring product has a different molecule attached to the side chain. The Merck product has a different thing. The Organon product has a different thing. There's a slight difference. Not a huge difference, but there's a slight difference. But we keep on changing it. Second thing is, then we combine urinary products with recombinant products. There, the urinary product is completely different from a recombinant product, even though it attaches to the receptor. That's the first thing. Once we have a standard dosage, then the next thing is the role of hyperresponder, normal responder, and a hyperresponder should be thought of. There is a subgroup of people who have everything normal, but they respond poorly with a given dosage in spite of good antral follicles, good AMH levels. That's a subgroup. These women, if you look at them closely, they have perfect regular cycles. You see antral follicles, but they don't go at the required speed they're supposed to go. But when you do a scan, you see the follicles. The E2 levels will be lower. That is when most of the time we increase the FSH dosage, but we are supposed to add LH dosage because that is the subgroup who are associated with LH receptor polymorphism. Here, if you look at it closely, most of the time they have regular cycles. And if you look at it closely, they might have slightly shorter cycles than usual. They might say, my cycles are 25 days. If you ask them much more deeply, 5 years back, how are your cycles? Then they say, my cycles are 30 days, they move to 25 days. This is a subgroup where midway, without LH polymorphism, you can add LH supplementation of 75 units to rescue that cycle. And in our study, that was the group we studied the LH polymorphism and then found out in that group, LH-CGR G by G is more in that group. And then if you supplement LH 75 units, then the cycle goes back normally. One need not do selectively. Either they have to do it for everybody because of pharmacogenomics so that they get experience. Because selectively, you can never pick up a hyperresponder. Only after the cycle is over, you can pick up a hyperresponder.
Yeah.
That's a fact. We don't accept that fact. And there is no role for selective usage of receptor polymorphism. The second thing is,
I think we can pick the hyperresponders from the stimulation also. During the stimulation also,
during
we
the
can know that.
stimulation, but your turnaround time to get that report takes time.
Like around 5 to 6 days, we come to know that this patient is responding hyper way,
True,
or we
but,
can go down.
But there you can't go and do the test there, no?
Yeah.
Yes. And also, like Dr. Ramraju was saying, there is a subgroup which does not display that they are hyperresponders. So that is also another thing that we have to keep in mind.
So either we do it for everybody or we don't do it at all. And with time, as the cost of the genomics are coming down, there'll be a time when the test becomes efficient. The second thing is, most of the authors who have published it are not using this polymorphism in their practice. Because when you look at the the Spanish and the Vietnam study. It was a joint study between two countries, and then they used 150 units for a period of many years. There is a lot of selection when you look at the number of cases. The second thing is they use it in the best patients. When you're using it in the best patients, there is no role for pharmacogenomics.
Absolutely.
So that is the limitation. Can you go back to the slide, last but one slide, where you're saying FSHR 29?
Sir, but at this
Which one, sir?
FSHR 29.
Last one.
No, before,
before.
yes.
The
This is
Delphi
the most
consensus or the
important. This is the most important statement in the FSH receptors that are currently being used. The reason is, go back.
29 variant. Yes.
Yeah, because the promoter region is present in this place, FSHR 29. And that is where the mRNA is being produced in the manufacturing of the receptors. And there, the number of receptors in the granular cell are lesser, so they need higher amount of gonadotropins, and they also produce fewer oocytes. But most people study only 616 and 615.
165.
So of the three receptors, if you look from a genetic point of view, this contains the promoter region, and this is the most important. And also we found the length of the stimulation also makes the difference in the number of mature oocytes in the lesser number of oocytes. Then next, go for the previous cycle where you compared 616 and 615.
The previous paper, sir, the polyodes and the
Any
three.
paper. Here what happens is, here 616 and 615 are in a equilibrium. You always find the same set of data except in small South African population. So 616 and 615, the A by A and G by G will match 99.9%. In our experience, we never got a difference between 616, 6166 and 6165. Only South African group is there which shows a difference. So when you look at that way, then the data is not matching to that statement. Because when they're in disequilibrium and both the things are same, your
Sir,
data should not be different.
sir, just a
The
second. I'm sorry.
strong linkage disequilibrium means both are one patient has this, that patient will have that also. I mean, if he has RS6166
Himanshu?
defect, he'll have
Yeah,
6165
Dr.
defect also.
Manjushri, just a second.
So
Himanshu, can you
when
please
look at-
enable chat for everyone? People are wanting to post the questions, but they are not able to post the questions. Please enable chat for everyone, or you can unmute everyone. You
if
can
you look at
unmute
Chat is
everyone.
enabled. No, chat is enabled now for everyone, I think.
Chat is enabled, okay. Or
Yeah.
you can unmute also, people can ask question. Himanshu,
If you-
please do that.
if you look at statement 3 and 4, they're contradictory. I mean, if you look at this statement 3, RS616, if RS616 has higher prevalence and hyper response, then RS6165 should also show that.
Yes.
Because of the strong
So-
linkage disequilibrium, no, sir? I mean, we are
Yes.
confused about that. That's why I'm
Yes.
asking.
So this data, because I spoke to Nicholas when he presented the data, in Spain. But what they said is, this is what we got. I accept that. But when you look at from a genetic point of view, the second statement, FSHR variant RS6166, higher prevalence of hyper response. So this should also be RS61665.
So, if I would like to explain the
What's your
disequilibrium
opinion,
thing.
Dr. Anuradhan?
Yeah, so I'll just explain the disequilibrium thing a little bit.
Yes,
Basically,
ma'am.
when you have what we have on the genetic code, it's called a loci. A particular point is called a loci. Now, of that particular point, you can have two different bases or variations. One is called a wild, the other is called a variation. So, if 6166 has a wild genotype, then 6165 should also have a wild genotype. That means the reference genotype, which has to be present on the genetic code as a reference or a normal or a wild genotype. If 616 is exhibiting a variation, then 615 will also exhibit the variation. That is why they are in linkage disequilibrium, that it is called. So if this changes, this will also change. If this doesn't change, this also does not change. So it is really intriguing to see how they found a significant difference in 6166 and did not find the same in 6165. In fact, there are papers, recent publications which say that if you do 6166, then there is no need to actually test for 6165. So they are
I
so.
totally agree with Dr. Anuradhan.
But
Yes.
sir, here in this forum, we want to give a take-home message to our youngsters. So there is a question somebody is asking that should 75 units of LH be added after gene testing in next cycle? Maybe you do a cycle, see the response if the patient gets pregnant. Because sir, IVF itself is a costly thing. The patient goes through a lot. Both the ways it is right, if the patient doesn't get the result, then you have to do the testing, or should we be doing the testing beforehand?
I would say this, of all the FSH and LH receptors, we have been doing since 2012, and we are using it routinely because we have an in-house sequencer. And we feel LH-CJR has a lot of clinical significance, and that too LH-CJR 312G by G needs LH supplementation, and that too 75 units. That is one thing. And that's the second thing is, if you don't have LHCR sequencing, how do you identify the pattern? If you look at the work from the NHS work, where there's a review article on LH supplementation, these patients with LHCGR 312G by G with adequate FSH dosage, you see adequate number of follicles, but the rate of growth of follicles, which should be 1 to 1.5 millimeters per day, does not happen in that way by day 6 or day 8. And the E2 levels are lower. In those patients, if you do LH polymorphism, 80% of the time you find LH-CJR G by G. And in that case, you can add LH supplementation. And that is how we initially started the work. Now we routinely do it and start it from the first day when there is LH-CJR Q12 G by G. Regarding FSHR, the data is too complex, and we have been using only RS394205, where we increase the dosage on and above the existing dosage based on the age, BMI, AFC or AMH, and the ovarian surgeries. And then on top of it, we add another 25 units, but never cross beyond 300 units. So the carry home message is, from whatever work we did, is in LH polymorphism, just because only Linderman and we did the work on LH, the Delphi consensus were very hesitant to give it into the consensus that LH recommendations. But when it comes to FSH, people have used no standard dosage. People have given, studied individual parameters, like one group studied 6166, one group studied 6165. and one group studied 1394205. In the process, each person was giving an advice. So it ended up with a confusion. And when you look at Delphi consensus, that's the reason you see a lot of disagreement in that consensus statement, because the problem there was in the field of pharmacogenomics, you are supposed to use it for every patient, not when you like it. That's my personal opinion.
Ma'am, can I ask a question?
Yes, please, please, yes, please.
We have so many
So,
questions in the chat box. I think
sir, what is your experience of, using recombinant LH is sometimes costly. What is your opinion of using HCG instead of recombinant LH, low dose HCG?
HMG. You mean HMG, not HCG. HMG.
No, HCG,
low dose,
HCG,
100 unit
low
HCG
dose
daily.
HCG, 100
Low
or
dose,
200 units daily.
100 or 200. Yes.
If you look at Marcos Filcori's work, which was done in 95, he used to give dilute the HCG ampule and give 37 units of HCG, which is equivalent to 100 units of LH. That's the Marco Filcori's work. But because I happened to give a series of talks with him in Europe, and that was in 95 when no polymorphism data was existing. And when I spoke to him about the use of LH, at that time, LH was not available for him. So if you read some of the Marco Filcori's work, you can use it because he got a decent pregnancy rates from his publications. But
Dr. Radha, you want
if you're using HMG, then there is no role for using, because he was using pure FSH, not HMG.
Yeah, I was thinking maybe HMG is better. Yeah, Dr. Anuradha, you wanted to say something.
Yes, I just wanted to give a small overview of what Dr. Manjushri has presented and my comments about these three papers, and also some take-home message, if possible, from a geneticist point of view. The first comment that I would like to make, especially about Dr. Nicholas' paper, is that there were no Indians included in that study. So when they say Asian, there are very few. In fact, I wrote to him and he did reply, kind enough to reply, that they did not have any Indians. When Asian means it's mostly Vietnam. Now, what we all have to understand is that the polymorphisms are ethnic specific and they can vary from one population to other. In fact, I have data like around 10,000 patients that we have done, and we found that there is a difference between state to state also. Okay, because we are so, there is an inbreeding and it is present. So therefore, the ethnicity, especially if not the state to state, at least the Indian ethnicity and the Indian publications have to be available to consider whether this is a common polymorphism in our state, country, or if it has any clinical correlation. That is number one. Then that was, I thought, was the limitation there when we talk about that paper in respect to our country. Second thing is about the linkage, which we have already discussed, FSH R variation. And we have actually published a paper where we have shown that the FSHR has a very clear poor response, and statistically in Indian population, it is showing that there is poor response. Again, same, we have also used the same 150 units starting dose and things like that, so that it was a very controlled study. And we are in the process of publishing another 1,000 patients data of live birth only on FSHR. So there is a strong correlation with FSHR polymorphism. As far as LHCGR polymorphism is concerned, Dr. Ramraju has done great work, sir. I can substantiate that with much more work that we have done on a large population studies that we have taken up on LHCGR. LHCGR, along with the variant that is commonly studied, we found a new variant that is associated with poor response and in idiopathic infertility. So we also include that. And because there is so much available of, I mean, as Dr. Ramraju was also mentioning, as the genetic studies and genomic studies become more and more available and they become more and more economical, which is happening right now, we will be looking at a lot of genes and a lot of polymorphisms. We published a paper on Indian population where not just FSHR, but AMH, AMHR2, FSHR, and LHCGR are contributing to poor or suboptimal response of LHCGR, ovarian response. So this clearly shows that response to stimulation is a polygenic condition. It's not based on a single gene. You can't just look at a single gene and say, yes, this is a poor responder. Because it is a polygenic condition, more and more studies are happening, and we are, like last year I have published 8 papers, and this year there are 10 in line with much more data, where we have to actually show what is called a polygenic risk score. So multiple genes are playing a role. All these genes must be tested, at least the ones that have been correlated and published. And then we have to derive a risk, and that risk has to be followed up with a pharmacogenomic implication. So this is my small input on the subject. Please don't consider ovarian response related to single gene. Although FSHR is widely studied polymorphism, and many a times I also want to clarify here that people get confused with the FSHR gene is very, very important for reproductive development, and it can result in pathogenic mutations which cause premature ovarian failure. And those variations should not be confused or tested in the process of, so people do things like exome sequencing and all that, which is not part of this. If you find a pathogenic mutation there, it will be a lot of confusion. So those polymorphisms which have been clearly established and published on Indian data that they have an association with infertility condition and ovarian response, and also have a pharmacogenomic application, must be studied, and a polygenic risk should be derived instead of looking at single genes. This is my take on this subject. And I'm very happy that something like this is being discussed on your forum for youngsters, and we'll be happy to share any genetic information or questions that you may have. I'll be happy to answer.
Thank you, Dr. Anuradha. We have 3-4 questions more.
Yeah.
This question is, should Dr. Shraddha, should 75 unit of LH should
This one is we already covered. Let's go to the next one. What is the
Dr.
clinical
Harita?
significance?
What is the clinical significance of the LH receptor polymorphism in planning trigger for the subset of population with hypo-response?
Like she means to say, is there a role of polymorphism in HCG also, if you have to give the trigger in hypo-response?
The answer is no, ma'am.
No. Okay.
Okay.
So can we use biosimilars like folitropin beta if polymorphism testing is not possible and patient is hyporesponder in last cycle despite normal AMH and AFC?
Yes,
I think
ma'am.
answer should be HMG stimulation, right, sir?
Like basically, I mean, just repeat the question, ma'am.
The question is like, can we use biosimilars like folitropin beta if polymorphism
ma'am.
testing is not possible? Maybe if we have got a low response in one cycle, ideally I think we should go for HMG stimulation, go for human menopausal gonadotrophin stimulation.
So what
If
do
it
you
is
say,
not
Dr.
possible.
Ramaraju? What is your answer on this? Can we use biosimilar like folitropin beta?
I mean, one thing is we never standardize the products, ma'am. That's what I feel. We
Yeah.
keep on changing the products. We use only one product so that we understand the molecule better in different situations. And second thing is, if there is a poorer response, then we never go back and look at the scan images and look at when did the triple line start, when did the cervical secretions start, what is the rate of growth, and then we never look at did we have any difficulty at the egg pickup. These are some of the areas we never document in our records. That's my personal opinion.
And what
If there is any reform, there should be a consensus statement on what is the starting dosage and how to calculate a starting dosage. Unless that is stabilized, the role of pharmacogenomics will never come into play. Pharmacogenomics is only fine-tuning, not gross tuning. Unless we have a consensus statement about how to plan a starting dosage, then only the role of pharmacogenomics come in for fine-tuning. That's my personal opinion.
And what is the cost and which labs in India is doing these testing? Next.
I'm sure many labs are doing selected polymorphisms, which we also do. We do selected FSHR, LHCGR polymorphisms. We also have a larger panel which looks at all infertility related issues, including implantation failure and recurrent pregnancy losses. And regarding the ovarian response, when we do the larger panel, we use AI and we take clinical parameters and we calculate the odds ratio, and we give a risk saying it is 3x times poor response or 5x times poor response and things like that, because we consider not just a single gene, multiple genes are considered, and then we give a result based on that.
And
So,
average
Dr.
cost
Anuradha,
of response?
Dr. Anuradha, I would request you to send me 1 or 2 papers.
Okay.
As promised earlier, after this general club, I will be posting those in the group so that
Sure.
everybody can go through those papers and then they can take a
Sure.
decision.
I'll send. I
Because
have
it is
presentations.
becoming confusing.
A
Sure.
question by
I
Shilpa
have some
Singhal.
papers which are international and on other things, but I will specially give you those papers which are related to Indian population.
Yes, yes.
Question by Shilpa Singhal, where to get the gene testing done in Delhi and average cost?
That I'm really not sure, but we offer testing throughout the country. If someone can contact me, and then definitely I can arrange sample collection and all that.
Ma'am, can I answer this question?
Yeah, Manjushri.
So I had, of course, Anuradha ma'am's Gene Tech also does, and Swagene is a company which also does this testing, and these labs are, and also Sir's place also the testing is done, Ramaraju Sir's place, and I guess the average cost ranges between 7,000-8,000, but the TNT is, as Sir told, it's at least 6-7 days.
Yeah.
Yeah,
If I'm
it's the same here. 7,500 rupees for selected panel and around 20,000 rupees for the whole panel with odds ratio of polygenic risk score. Same thing.
Okay. I think this was a very interesting topic and a never-ending one. We can continue on and on and on, and there will still be more information needed because honestly speaking, I am still confused. At the end of the whole discussion, there is something else going on in the mind. One thing I would like both of you, Dr. Raju and Dr. Anuradha, to summarize in 2 sentences. Number 1, who should do? Number 2, if this is not possible to be done, then what should be done?
The first thing is, if you are going to do it for everybody or no.
Okay.
The
Yeah.
second thing is, what was the second part of the question, ma'am?
If it is not possible to be done because of financial or whatever.
Cannot afford to do that.
Basically, I'll be able to tell about LH polymorphism, how to pick up without doing an LH polymorphism.
Yes, please tell.
With adequate dose of FSH, with adequate number of follicles, if your follicles are not growing at the rate by the 6th day or 7th day, and if you are using FSH molecule, then there is no need to increase the FSH molecule, add LH 75 units. That's the carry forward message, what I could get in this 10 years.
My answers for these 2 questions.
Yes,
Okay.
please, Dr. Anuradha.
Yeah, my answer to these 2 questions. First, I totally agree with Dr. Ramaraju that it should be done for everybody or not done for anyone. But given the practical situations that we all face, what I have seen is that most of the physicians who refer, generally to gene tech, they refer for the complete panel, not for the selected polymorphisms. But when they do that, even for selected, many of them that I have seen use it for cases with recurrent implantation failure or recurrent, poor response that they have already experienced, in one cycle, or maybe implantation failures or failures of IVF cycles, or poor response. So when they see this, or even when the oocyte quality is low, or in such cases, I see that 80% of cases that come to us are already with some background experience of poor stimulation in some form or the other, or failures that are referred to us. So that's one maybe, if not for all, then you could look at recurrent implantation failures or poor
At least
response.
after one cycle failure.
Yeah,
Like
maybe.
I want to add one thing.
Sure.
There is no role for receptor polymorphism in poor ovarian responder with low AMH and low AFC.
Yes.
The role is in a normal responder or in a hyper responder or an excess responder is the receptors, but not in poor responders with low AMH and low AFC. There is no role there, selective role of receptor polymorphism. And people think receptor polymorphism is for that group, and it is not for them at all.
So especially not FSHR in those cases, but there are other genes, sir, that are associated with poor AFC and low AMH. There are many genes. There are around 11 genes that contribute to that factor, and some of them could be AMH, AMHR2, and those genes have to be tested in those cases. And there are certain pharmacogenomic implications that are evolving even for those genes, which I think that will take another one full session to discuss those. So yeah, so that is one thing about who to refer. And what was the second question, Dr. Nisha? And that though I think Dr.
Yeah.
No,
my question is, like, suppose I think I have answered my question myself. If a patient has gone through a cycle and we have embryos and we get a failure, then there is no point in doing now, because we already have embryos.
Yes.
Only with stimulation only. Okay.
Yeah.
So
Okay,
that's
thank
a
you,
good
everyone.
take-home message. Yeah, thank you, Dr. Raju, Dr. Rama Raju, and Dr. Anuradha. So it's a good take-home message that it's not for the patient with poor ovarian reserve or with the low AMH. And as far as we know, with the poor ovarian response or low AMH, we already use HMG or Minopure or anything like this only. So thank you a lot. Thanks a lot. Thanks for
And
sparing.
thank you. I congratulate Manjushri for selecting such a nice topic and having such a wonderful discussion.
And bringing it to forth.
Thank you,
I
everyone.
would like to thank you,
Ma'am.
IFS, for giving me the opportunity, ma'am.
Yeah, thank you all, Young Turk, for your presentations, and you everyone here. And I request those still not the member, become the member of IFS. It hardly 2 minutes job to become the member. So thank you very much. So we'll meet now on the 4th Friday of the month.
Thank you, ma'am. Neeru, ma'am, you have always been a big support, and I know I can rely on you anytime, whenever. So she's always the backbone of everything. Thank you, ma'am. Neeru, ma'am. Thank you, and thank you, sir, and Shweta, ma'am.
Yeah.
Thank you.
Thank you, sir.
Thank-
Thank you, Dr. Anuradha. Thank you, Dr. Rama.
Thank you, ma'am.
Thank you, Dr. Anuradha. Thank you, Dr.
Actually, you
Rama.
have done a good job to the topic.
Thank you.
Okay, okay. So we encourage other participants also to come forward and present your work. Thank you. Thank you.